Hello, today I will be presenting on epigenetics and Angelman syndrome. So we know that epigenetics are when changes occur in gene activities due to environmental factors like lifestyle choices that can cause them like smoking. And these don't change the DNA sequence. Examples of this are DNA methylation and histone modification. So these changes will affect gene expression. And for example, DNA methylation, which I will get a little bit more specific later on in this presentation, it occurs while DNA is replicating. That's when it gets affected. So you can see the central dogma. We know that genetic information gets passed down from DNA to RNA to protein. So typically when the DNA is replicating itself, that's when DNA methylation will affect it most. So on the left, you can see a diagram that I grabbed from the textbook where we know there's a methyl group added to our DNA bases. And I chose this topic because during the semester, I thought the epigenetics and chromatin module was the most interesting to me. So I wanted to look into it further and research disorders that happen because of this. So Angelman syndrome is a genetic disorder, and this causes mental disabilities. It causes balance and motor issues, as well as seizures in more severe cases. This occurs when one copy of the UBE3A protein, so this is the ubiquitin protein ligase E3A, in the 15th chromosome, and it gets passed down from one parent to the offspring. So typically, because the other parent's DNA got silenced, or sorry, the other parent's gene got silenced. So this is a great example of imprinting. A couple facts are that 70% of patients have deletions in chromosome 15 from the maternal allele. So this is the most common genetic situation that they have going on. So 7% have paternal uniparental disomy, which means that the offspring will receive two copies from one parent. And 2 to 5% are caused by epigenetic defect or DNA methylation, which I thought was very interesting because this is a bit more rare to find. And on the right, you can see a diagram of the phenotype of people with Angelman syndrome. During my research, I found that there are 5 molecular classes of Angelman syndrome, and as it says here in the title, with methylation status. So what that means is that from this article, I discovered that classes 1 to 3 have a loss of methylation, and classes 4 to 5 have a normal differential methylation. So in this study, they did an assay of the allele-specific methylation of a SNRF, small ribonucleoprotein N gene, exon 1 DMR. So that was the name of it. And they did this to be able to differ between the type of classes from Angelman syndrome. So this is done to sort of diagnose the type of mutation or the genetic abnormality that the patient has going on. So genomic imprinting plays a very big role. We briefly went over it in my first slide. So imprinting will determine the origin of the chromosome from the parent. So we know that one gene can get silenced by DNA methylation, and this occurs typically after the DNA gets synthesized during DNA replication. The specific base pairs, they can get methylated, and I mentioned this already. And again, this will occur because of environmental factors or mutations during development. So what do these methyl groups do to the DNA? So they block protein binding that, proteins that will facilitate transcription. They can also bind to proteins that will promote this further compaction of DNA. It will promote more compaction that will result in transcription from being prevented. And usually if the area near a promoter gets methylated, then it will for sure repress gene expression, because we know that methylation can either activate or repress, and depending on where it's methylating, so if it's near the promoter, it will repress gene expression. And where does this occur? It occurs in the CPG regions, which are the cytosine, guanine, those phosphodiester bonds, when they are on the same DNA strand. So down here in this figure, we see that this is a diagram of this study that I looked into. And the blue regions are paternal alleles that are expressed, and the gray regions are the maternal alleles that got silenced. And the circles, the white circle and the paternal alleles, the paternal DNA, it was not methylated. And the maternal alleles, where the black circle is located, that indicated methylation occurred. So that affected gene expression. That's why the maternal DNA got silenced. So the loss of the UBE3A gene results in Angelman syndrome. So UBE3A, this gene is a co-activator and will enhance transcription. So when this gene is lost, transcription can't occur. So in this experiment, they inactivated UBE3A in mice from the maternal DNA, and they noticed that Angelman syndrome symptoms were present. So these proteins in this Western blot, the UBE3A, the ECT2, and the AR-GAPDH, AR and EC2 are known to be regulated by UBE3A. And in this Western blot, we can see that there is abnormal expression for EC2 and AR for the brain. And this is caused because of the UBE3A, the loss that happened. So because there was no UBE3A, and these proteins that UBE3A regulates were not regulated anymore, then this resulted in Angelman syndrome symptoms being present. And these are my references. Thank you.